Hello, today we shall study something about plant tissue culture. You might have heard about this plant tissue culture as a branch of biotechnology here and there, especially in the news media in recent times. Let us try to understand what are the features of this a very promising field of biotechnology called plant tissue culture. See, plants propagate basically in two methods. You take any plants, either it is propagated or it is cultivated by using its seeds or its cuttings. For example, if you have a mango tree, you have a jackfruit tree, they are all propagated by using their seeds. Otherwise, if you take a rose plant or a jasmine plant, they are propagated by using their cuttings, either root cuttings or stem cuttings. So basically we have two modes of propagation of any given plant. But often what happens in our day-to-day life, maybe in agriculture or horticulture or any other form of enterprise which needs the cultivation of plants, is we have a cultivation problems in one or the other plant. For example, if you might have seen the very beautiful flowers of the orchid plants, but these orchid plants are neither propagated through seeds nor through their cuttings. So it becomes very difficult if you want to propagate orchid plants in a large scale. Likewise, if you want to cultivate a rose plant, of course you can cultivate that plant by using their stem cuttings or root cuttings, but often the desired quality in the quality of the product, maybe the rose color or rose flower shape, rose flower fragrance may get reduced if you keep on cultivating that plant through their cuttings. So therefore, often in agriculture we face problems for cultivation of plants by using seeds or cuttings. In that case, we need some other forms of cultivation techniques which can ensure the real quality of the original plant material. It can be achieved by using plant tissue culture. All these experiments started nearly 100 years back, but it was a very laboratory based experiments. But in last 30-40 years, this has been brought to the field. I mean, this technology is being widely used in most of the plants across the globe, wherein plants are being propagated through the tissue culture techniques. Now let us see what are these techniques. See, plants are composed of basically cells and tissues and then organs and the whole plant. Say for example, if you take a mango tree or a mango plant, you have different tissues. Maybe the bark is a tissue, or any given content of the leaf or a fruit or a root is a tissue. Then the organs, for example, a root is an organ, or a bark is an organ, stem is an organ, leaf is an organ. Then the whole plant. So plant is organized, or it developed in different levels of organization. So we can actually culture these plants in all these levels. Suppose if you want to get only the tissues of a plant, that can be cultured by using tissue culture technique. If you want only the organs, maybe you only want leaf, or you only want fruit, or you only want leaf or a flower, that can also be cultivated in the plant tissue culture lab. And of course, the entire plant can be propagated in the tissue culture lab. Thus, tissue culture provides you a technique of getting a part of the plant or the whole plant. Then how it is done? Basically, plant tissue culture technique is a mimicking the real growth pattern in the nature. For example, if you look at any plant, it is fixed to the soil, it absorbs the nutrients from the soil, and it takes up the water, minerals, etc. from the soil, and then it makes use of carbon dioxide and the oxygen from the atmosphere, and it makes use of the light from the sun. So by using all these components, both from the sky and from the soil, plant makes its own food and it gets its nourishment. Same thing is mimicked in tissue culture laboratory in the lab. Here, a nutrient medium, it's almost like a food. It is prepared by making use of many different chemicals in the lab, which provides all the nutrients for a plant which are supposed to be taken from the soil. And the light is provided by making use of electric light, and the temperature and the humidity are maintained whatever is required. So in a tissue culture lab, there are three things basically essential. One, the right mixture of nutrients in the form of tissue culture media that is prepared out of synthetic chemicals. Second is the controlling the environmental factors like temperature, light and the humidity by making use of different equipments. And the third one is maintaining very aseptic condition. Aseptic condition means free from any microbes, because plants are vulnerable to different microbial infection. Any given plant may be attacked by a fungus or a bacteria or a virus, so a tissue culture lab should be free from any those kind of infections. So if you maintain these three, so then you can pick up a small piece of a plant as an original source. Suppose if you want to cultivate a mango tree in a tissue culture lab, what all you need is a small tip of a mango leaf, or a small tip of a mango root, or a small part of a stem. A small piece is enough. Then it is brought to the lab, it is put into a glass vial and the nutrient media is provided and the necessary growing conditions are maintained. So within a matter of say 2 weeks or 3 weeks or 4 weeks, you will see that this simple piece of a plant starts growing into root and shoot, and then it becomes whole plant. So it's a marvelous thing to see a simple a plant part from the nature can become the whole plant in a laboratory condition. This technology is being used in various forms of applications in today's industry. As I said earlier, the first of all, the purpose of plant tissue culture is to get a whole plant. Suppose if you have any rare plant, if you want to cultivate any rare plant, what you need to do is you simply bring a small piece of a plant from any corner of the world, and then we can bring it to our lab, and then we can grow it into a whole plant by making use of the media available in the lab and the growing conditions available in the lab. Second part is you can go for only the production of only one organ or one part. For example, if you are interested in a very rare flower of a plant, it is possible to produce a only flower in the lab, or only leaf in the lab, or only root part in the lab. Today in many herbal drug industries, many pharmaceutical industries, people are using this technique for producing the required organ. They are not interested in the whole plant, but they are interested only in the only required part of the plant. And the third dimension could be, third approach could be developing or the producing certain chemicals in a plant by using tissue culture method. Again, in many pharmaceuticals, actually most of the plants actually provide the drugs because of there are some chemicals. Suppose if we are able to produce those chemicals in the lab by using tissue culture, directly we can use them in the drug. We need to go for production of entire plant. Thus, plant tissue culture can be used for the production of whole plant or its part, or even some of its chemicals which are used in different industries. Finally, why should we go for tissue culture when we are getting the plants in nature? This is a very valid question. A tissue culture is a technology. It requires huge investment. It requires different kinds of technology. So that's why for a routine propagation purpose, we may not go for, or we need not to go for tissue culture. But wherever there is a problem in natural propagation, whenever there is a problem in seed germination or a propagation from root cuttings or stem cuttings, then we can go for tissue culture technique. Or if you want to preserve some species, plant species for long in gene banks artificially, you can preserve them through by using tissue culture techniques. Or in industries, we can always go for the production of certain parts of the plants or the chemicals of the plants by directly growing them in the lab. Thus, this plant tissue culture, one of the promising fields of biotechnology, is being used across the globe in present day agriculture science, in pharmaceutical science, in conservation biology, in environmental science, and likewise. So in many fields this is being used. It has got huge opportunities in terms of their employment opportunities also. The students who are trained in tissue culture in India are getting placed in different parts of the globe, in different industries, those who are using tissue culture as a technology. Thus, it is considered as one of the important technology of modern day biotechnology. Thank you.